In this video, we're going to take a look at the process of transcription, which is basically where cells are going to take the information that is stored in their genes on the chromosomes and copy that into RNA molecules. And some of those RNA molecules can actually be used to synthesize specific proteins. So what we're looking at here is our prokaryotic chromosome, again, that can synthesize proteins that are out there. And like I said, when we look at this process of transcription, we're basically looking at the cell making a copy of a short segment of the DNA. And that short segment of the DNA is what we often refer to as genes. And these genes code for a functional product. And when we look at these functional products, they are RNA molecules, which are just copies of that segment of the DNA there. Some of the different types of functions that we can see for these instructions that are stored in genes, one of them is known as a ribosomal RNA or rRNA, and these molecules are used to actually form the framework for a ribosome. And a ribosome is a big enzyme that is made of RNA and proteins that's used in the process that we look at after transcription of translation. Other of these genes will encode a functional RNA known as a transfer RNA, and the whole function for a transfer RNA, as you can see over here, is it's going to actually be delivering amino acids to the ribosomes for this process of making proteins. And the last type of function that we can get from a gene is a messenger RNA or mRNA, and these short little pieces of RNA are actually going to carry the information that goes out to the ribosome, and it directs the order of the amino acids that are actually included in your protein that is synthesized by the ribosome itself. And so if we look at this overall process for transcription here, we can break it up really into three different processes that are taking place during it. And if we look at this first process over here, what's known as initiation, this is where an enzyme known as RNA polymerase, similar to what we saw for our DNA replication, but in this case the enzyme actually makes RNA polymers here. This enzyme's floating around in the cells, and it's going to bind to the DNA, and it runs into what's known as the promoter sequence. And this is basically the start site for transcription. The RNA polymerase itself has its own helicase activity, so it will be able to unwind the DNA at the promoter, and then effectively what the RNA polymerase is going to do is going to break those hydrogen bonds, and it's going to start doing this process of transcription, and that's where you're making an RNA copy of your DNA there. And if we look at the middle part, what's known as elongation, here you can see the complementary RNA bases are being attached together to make our piece of RNA right there. And if you remember, your DNA strands are anti-parallel. They're going in opposite directions. So we see 5' to 3' there and 5' to 3' on the bottom going in the opposite direction. And this RNA polymerase is using one strand, and it's making a complementary copy of what's known as the template strand there. So again, if you have your DNA and it has A, C, G, and T, when you're looking at your RNA, you're going to have those complementary bases. So an A in the DNA is going to complement a U in the RNA, and likewise a C in the DNA would complement a G in the RNA there. So this elongation phase is basically the RNA polymerase just coming along, unwinding your DNA as it moves along that segment or gene right there, and it is attaching the complementary RNA bases to make your RNA molecule. And then at the very end of this process, your RNA polymerase runs into a set of bases on the DNA that are known as the terminator. And effectively what goes on here is your RNA polymerase is going to stop making a copy of the DNA. The RNA polymerase falls off and it releases your RNA. And remember, from the beginning, those RNA molecules could be ribosomal RNAs, and then they're destined to serve as the framework for your ribosome. Some of them will be transfer RNA molecules, and those are the ones that are going to get charged up with amino acids, and some of them are messenger RNA, and those are the ones that are going to actually get used during that process of translation that we'll look at in the next video.